Victor for the nice introduction, and thanks TDT for put together this event. I thank all of you come to the talk during the election time. So today I will share with you our recent work on the neural circuit and sleep homeostasis regulation. So as we all know, so the sleep is a conserved phenomenon across evolution, not only in mammals, but also in birds, fish, insects. Even the jellyfish and worm has been reported to have sleep-like phenomena. So this conservation across the evolution suggests that sleep must serve some very important functions. However, right now, we still don't have a clear idea why do we need sleep. It is now generally accepted that the real secret of sleep probably in our brain. So it's our brain needs sleep. So in order to know why do we sleep, I think we need to at least understand two fundamental questions. First, what is the ultimate function of sleep behavior? Second, how does the sleep-wake cycle be regulated? Today, I will share with you our work on the regulation part. So on the study of the neuromechanism of sleep-wake regulation, there are generally two schools of thoughts. So one group of researchers try to find what kind of neural activity in the brain, which structure control the transition from sleep to wake, sleep-wake transition. The other group of scientists trying to identify genes and molecules important for the sleep-wake behavior. So today, my talk will also focus on these two parts. In the first part, I will talk about the neural circuit. In the second part, I will talk about the sleep homeostasis regulation. So the brain region we have been focused on is called basal forebrain, or BF for short. So the basal forebrain is part of the ascending activation system, which is the key system to maintain our wakefulness. So the BF is famous because it contains a large number of cholinergic neurons that project to entire cortex like hippocampus. So if we do manipulation to the basal forebrain, for example, activation or lesion, you can basically bidirectionally affect the sleep-wake cycle. On the other hand, the BF is also intensively studied in the cognitive neuroscience, because one study has shown that in patients suffered from the Alzheimer's disease, there's significant loss on the number of cholinergic neurons in the basal forebrain compared to the control patient. So from this brief introduction, you can find that on the basal forebrain, a study on the sleep-wake regulation and cognitive function, people have been focused on the cholinergic neurons. However, the cholinergic neurons is really only a small part of the basal forebrain. There are also large numbers of other cell types, like the glutamatergic neuron, which can be labeled by the VGLUT2, or large number of GABAergic neurons. So the GABAergic neurons is known to be diverse. It can be further divided into the power-booming positive one and the somatostatin positive GABAergic neurons. So a few years ago, when we started the basal forebrain project, the simple question we asked is, how do the four genetically defined cell types contribute to the sleep-wake regulation? So we asked four questions. First, what are the firing patterns of different BF cell types during sleep-wake cycle? Is there, for example, is there a cell selectively active during wakefulness, or is there a cell selectively active during sleep? Second, how does the cell type specific manipulation affect sleep-wake cycle? And what is the local and long-range interaction of these four types of neurons? So for the first two questions, we use optogenetic methods to perform the cell type specific recording and manipulation. So what we found is that a majority of neurons in the basal forebrain, including the glutamatergic neurons, CHAD neurons, and PV neurons, are so-called wake neurons, which means these cells are more active during wakefulness. If you activate this cell, it will increase the probability of wakefulness. So here's an example of wake active cholinergic neurons. We use the EEG and EMG to define different brain state. Different brain state is color-coded over here. So you can see the wake is labeled in the gray. During wakefulness, the cholinergic neurons firing rate is generally very high. So this neuron is also active during the REM sleep or rapid eye movement sleep. But during the non-REM sleep, the neuron is very silent. So this finding is basically consistent with published literature that the basal forebrain is a wake-promoting brain regions. However, we also observe some neurons show opposite firing pattern during the sleep-wake cycle. Here's an example. You can see during the wakefulness, this neuron is very silent, but instead it's very active during the non-REM sleep. Interestingly, we only found this kind of sleep neurons in the somatostatin positive GABA-2 neuron groups. If we use optogenetic method to activation of the SOM neurons, we can increase the probability of the non-REM sleep. So next, we ask how these four kind of neurons interact inside the basal forebrain. So we use the optogenetic assisted brain size recording to map all the pairwise connection of the four cell types. Interestingly, we found all 3 weak cells receives intensive inhibition from the sleep-promoting SOM neurons. We think this might be the reason that SOM neurons promote sleep. So the SOM neurons can basically inhibit all these weak neurons to promote sleep. Last, we ask, what is the interaction of these 4 types of cell with other brain regions? Because we know in the brain, none of the regions is isolated. They all receive a large number of input from other brain regions and send projections to a lot of other brain areas. So to answer this question, we use standard virus tracing method, and we get the whole brain input and axon distribution of all 4 types of neurons. Interestingly, we found all 4 cell types receive input from similar set of brain regions, including the striatum, hypothalamus, pallidum, and olfactory regions. And also, we found these 3 weak neurons seem to form 2 distinct output pathways. The cholinergic neurons mainly innervate the cerebral cortex and hippocampus. On the other hand, the weak GLUT and PV neurons send heavy projection to the subcortical regions, including the thalamus, hypothalamus, and midbrain regions. So it's interesting because all 3 neurons are weak active neurons, and if you activate them, you will promote the wakefulness. But their downstream target was very different. This result suggests that you can have multiple parallel pathways to promote wakefulness. On the other hand, the sleep-promoting SOM neurons send heavy feedback inhibition to the common input region of the basal forebrain. This may be another reason that the SOM neurons can promote the sleep. So to summarize the first part, in this part, we performed the cell type specific comprehensive analysis of the basal forebrain neural circuit for the sleep control. So in the function part, we found all the cholinergic neuron, glutamatergic neuron, and the PV neurons are weak and REM active. If you activate them, you will promote the wakefulness. On the other hand, the SOM neurons contain some sleep active neurons. If the activation of SOM neurons can promote sleep. And we also performed a comprehensive map on the long-range and low-cost synaptic interaction of the BF cell types and revealed some interesting properties of this cell. So the first part is mainly work I performed when I was postdoc in Yang Dan's lab in UC Berkeley. The second part, I will talk about the work from my own lab, study of the sleep homeostasis mediator adenosine. So generally, the current model of sleep regulation is the model proposed by A. Brobley in 1982, called two-process model. So the two processes in the model is circadian rhythm and sleep homeostasis. So the circadian rhythm represents a long-term adaptation during evolution to the external 24-hour day and night life cycle. So the sleep homeostasis really describes the most fundamental feature of the sleep-wake cycle. So during wakefulness, the homeostatic sleep drive gradually increase. When we reach a point, we will go to the sleep to decrease the sleep drive. So this phenomena suggests that the sleep promoting substance can accumulate during the wakefulness and cause the increased sleep pressure, eventually cause the transition from wakefulness to sleep. So this describes the most fundamental questions in the sleep-wake regulation study, is to find the biological nature of the sleep-promoting substance. So one of the theory is called adenosine hypothesis. So adenosine is the metabolite of the cell energy molecule ATP. So when ATP lose all the phosphate, it will become adenosine. So this theory, it is believed that during wakefulness, the cell metabolism need more ATP and then lead to the accumulation of the adenosine. The accumulated adenosine eventually will activate the A1 receptor and inhibit the neural activity and then increase the sleep pressure. So for the same reason, the coffee is widely used to maintain wakefulness because caffeine is an antagonist of adenosine receptor. It can block the adenosine-induced increase of sleep pressure. So this is the classical study from Dr. McCauley's lab at Harvard University, published in Science, 1997. So in this paper, they recorded the adenosine dynamic during a sleep-wake cycle in the cat Basal Forebrain using microdialysis technique. What they found is that during wakefulness, the adenosine level is relatively high comparing to that during the sleep. Interestingly, if you subject the cat to the long-term wakefulness, you can see that during the prolonged wakefulness, there is gradually increase of the adenosine, and the adenosine level gradually decreased during the following recovery sleep. So this kind of adenosine dynamic during prolonged wakefulness and recovery sleep matches very well with the proposed homeostatic sleep pressure hypothesis. So the authors then used pharmacological method to increase the concentration of the adenosine. Interestingly, they found only adenosine increase in the basal forebrain can lead to the increase of the slow-wave sleep. Instead, if you increase the adenosine in the thalamus, it causes no increase in the slow-wave sleep. So in the end, it is concluded that the basal forebrain adenosine is a physiological sleep factor that controlling the sleep homeostasis. So from the first part of my introduction, we know in the basal forebrain, different patterns of neuron activity control the sleep-wake cycle. So it is unclear how this kind of neural activity control the accumulation of the adenosine and also the sleep homeostasis. So to study the neural control of the adenosine, we need a better way to monitor the adenosine dynamic during the sleep-wake cycle. In the classical paper, 1997 paper, the authors can only get one sample every 10 minutes. This is way too slow compared to the neuron activity, which is typically at the scale of a millisecond. So we then collaborate with Dr. Yulong Li's lab at Beijing University, use the adenosine probe they developed recently. So this adenosine probe basically is a chimera protein from the human HOA receptor and the CP-EGFP. When the receptor combined with adenosine, it will change the structure. The structure change will lead to a fluorescence increase of the coupled EGFP. So basically, we can measure the fluorescence change to know the dynamic of the extracellular adenosine. So the new adenosine sensor has a very fast temporal resolution. So the onset dynamics, the kinetics is around 70 milliseconds. It basically can provide a real-time monitor of the extracellular adenosine. So use the sensor, we measured the adenosine level in the basal forebrain during the sleep-wake cycle, use this fiber photometry method. We can see during wakefulness, the adenosine level is generally high comparing to the non-REM sleep. So this is consistent with a previous microdialysis measurement. However, because of the fast temporal resolution of the sensor, we observed very fast dynamics of the adenosine instead of the slow dynamics observed using microdialysis previously. We also have the temporal resolution to resolve the adenosine dynamic during REM sleep. So the REM sleep here is labeled as pink. You can see during a REM sleep, there's a very large increase of the adenosine. So this kind of fast adenosine dynamics suggests to us that the adenosine dynamics is very likely to be neuroactivity dependent. So in the first part of the talk, we have known that there are 4 types of neurons in the basal forebrain, has distinct firing patterns and also different contribution to the sleep cycle. Then we asked, whether different kind of neuroactivity can have a different role in regulate the adenosine dynamics during the sleep cycle. Previously, the research suggests that the cholinergic neuron may play a role in controlling the adenosine dynamics. So we first studied the role of the cholinergic neuron. So we used this GCAM and the adenosine sensor to simultaneously record the cholinergic neurons activity and adenosine dynamics in the basal forebrain using fiber photometry. So here's an example. So the blue trace is GCAM activity from the cholinergic neuron. The green trace is the adenosine dynamics. So you can see the 2 signals are correlated very well. So this is the summary result. You can see when the GCAM signal is high, the adenosine signal is also high. So this kind of correlation is absent when we randomly shuffle the GCAM signal. More importantly, if we zoom in to check the time course of the 2 signals, we found that the GCAM activity, which is labeled blue over here, is always precedes the adenosine dynamic. So this result suggests that the cholinergic neurons activity maybe can drive the increase of the adenosine. To confirm that the cholinergic neurons activation can indeed drive the increase of the adenosine, we use the optogenetic method to activate the cholinergic neurons and record the evoked adenosine release using the fiber photometry. From this example, we can see that so after the brief laser application, we can see a significant increase of the extracellular adenosine. So this result suggests that the cholinergic neuron can indeed provide some contribution to the adenosine increase during the sleep cycle. However, we also see that this evoked adenosine level is relatively small. And also, when we apply the laser in different trial, the evoked adenosine, there's a lot of variation in the evoked adenosine release. So this result suggests to us that there may be some other cell types that provide bigger contribution in controlling the adenosine dynamics. So in our previous work, we have found that the glutamatergic neurons can contribute much more to controlling the sleep-wake behavior. So we hypothesize that maybe this neuron can provide more contribution to control the adenosine dynamics. So use the same recording method. We also see there's a good correlation between the glutamatergic neurons activity and the adenosine dynamics. Also, the activity from the glutamatergic neurons always precede the change with that in the adenosine level. More importantly, if we use optogenetic method to activate the glutamatergic neurons, we observed a robust increase of the extracellular adenosine. So this result suggests that the glutamatergic neurons can provide more contribution in controlling the adenosine dynamics during the sleep-wake cycle. So one important thing to note that we activate the chat neuron and vagal neuron using the in vivo measured firing frequency. So the difference in driving adenosine release during the optogenetic experiment can likely reflect the ability of these neurons in controlling the adenosine level in vivo situation. So to further confirm the role of weak group neuron in controlling adenosine dynamics, we performed the loss function experiment by using the caspase-3 to selectively lesion the weak group neuron in the basal forebrain. So comparing to the control animal with no lesion, we can see the adenosine level was significantly reduced. So together, this result suggests that the glutamatergic neurons play a major role in controlling the fast adenosine dynamics in the sleep-wake cycle. So last, we asked, what is the physiological significance of our finding? So according to the adenosine hypothesis of the sleep pressure, so the increase of adenosine during wakefulness can mediate the increase of sleep pressure during wakefulness. Given our finding that the glutamatergic neuron in the basal forebrain has an important role in controlling the adenosine accumulation during wakefulness, so we predicted that if we selectively lesion the glutamatergic neuron, it can cause a decrease of the sleep pressure during wakefulness, maybe can lead to more wakefulness. So we indeed measured the sleep-wake behavior of the lesion animal and the control animal during the 24-hour period. So we can see that comparing to the control animal without the lesion, when we lesion the weak group neurons in the basal forebrain, the mice sleep significantly more time during wakefulness during the dark period. So we interpret this result as that when we lesion the weak group neuron, cause a less accumulation of the sleep pressure. So the less, the lower sleep pressure leads to more wakefulness. We also measured the recovery sleep after prolonged sleep deprivation. So we subject the mice with 6 hours of sleep deprivation starting from the light on period, and then measure the recovery sleep. We can see compared to the control animal, the lesion animal has less sleep during the recovery sleep. And also, if we measure the decline rate of hourly sleep time, we can see that the lesion animal seems to have a faster decline. So the interpretation of this result is that the lesion animal can have a faster decline of the sleep pressure accumulated during the long-term sleep deprivation. So to sum up, so in this part, using a newly developed adenosine sensor, we found a neuroactivity-dependent rapid adenosine dynamics during the sleep-wake cycle in the mouse basal forebrain. When we optogenetically activate the glutamatergic neurons, we found a prominent increase of the extracellular adenosine. When we selectively lesion the glutamatergic neuron of the basal forebrain, we cause the adenosine level, we cause the low level adenosine, and also in the behavior level, we observed a significant increase of the wakefulness, which probably due to the decrease in the sleep pressure during wakefulness. So what is the significance of our work? So I think first, the significance is we found how the adenosine in the basal forebrain was regulated by neuroactivity in the basal forebrain. We found that there are a different type of neurons activity in the basal forebrain have distinct contribution to the accumulation of the adenosine. With the glutamatergic neuron has a much bigger role than the cholinergic neurons. Second, as I mentioned in the background introduction, there are generally two parallel research directions in the study of sleep-wake regulation, neural circuit and gene and molecule. So there's little crosstalk between the two fields, partly due to the technical the difficulties. So in our research, by studying the neural control of the metabolic small molecule adenosine, we basically eventually build a bridge between the two fields. So last, which I think is the most interesting implications of our research, we provide an answer to why do we sleep. We found that the wake promoting effect of the so-called wake neurons may come with a great cost. It can cause the increase of sleep pressure, we're stimulating the adenosine release. On one hand, we need this kind of neuroactivity to promote and maintain our wakefulness. On the other hand, this kind of neuroactivity will lead to the accumulation of the adenosine. Accumulated adenosine will cause the increase of sleep pressure, eventually trigger the transition from wakefulness to sleep. So this work is a collaboration between my lab and Dr. Yulong Li's lab at Beijing University. So the adenosine probe was developed by Italian postdoc in Yulong's lab, Wu Zhaofang, and the rest of the work was performed by 2 graduate students in my lab, Peng Wanling and Song Kun. So together with our previous work on study the local and long-range circuit of the basal forebrain, we provide a comprehensive neural mechanism of the basal forebrain for the sleep-wake regulation. So with that, I would like to thank all the lab members and our collaborators, especially Dr. Yulong Li from the Beijing University. So the adenosine probe they designed provide a basis for our research. I would also thank all the agents, the funding agency, and thank all of you to attend this webinar. I'm ready to take questions. Thank you.
Thank you so much. That was fascinating and very detailed structure analysis of the sleep cycle. So we have our first question. Which type of basal forebrain neurons express adenosine receptors?
Yeah, that's a very good question. So currently, I think we don't know the exact answer. What I can know is previously there are some indirect evidence, which when I say it's indirect evidence, basically it's pharmacological blockade of adenosine receptor. And it shows that a lot of, for example, the CHAD neurons and PV neurons, I think they all have the adenosine receptor. This represents another research project we kind of currently perform in our lab. So we want to use the single cell RNA-seq to comprehensively analyze not only the adenosine receptor, but also other receptors, for example, neuromodulatory receptors, to know how these kind of receptors express on different kind of cells. Yes.
Let's see. If nobody, does anybody else have a question? If not, can I ask a quick question about this? So, sure. Do you think that the adenosine accumulation is a byproduct of wakefulness, or do you think it is used as a way of promoting the circadian cycle?
You mean the sleep cycle?
Yeah.
Yeah. So one hand, I think you can see it's a byproduct because it's so all the neural activity like the spiking or the cell signaling, all kind of thing you need ATP, right? When you use ATP, basically you will generate a byproduct. One of the byproduct is adenosine. I think in this way you can see the adenosine is a byproduct. But on the other hand, so you can see that this kind of mechanism is has a very long time evolution. During evolution, I think our system maybe can use this kind of byproduct to do some good things. I think one good thing is they can use adenosine to activate the A1 receptor, basically provide a feedback inhibition of the neural activity. So when the system is too hot, you need too much energy, then you can generate a lot of adenosine. But adenosine basically can serve as a brake to inhibit the system, to let the system not get overhot. Yeah, that's my understanding. Yes.
Okay. We have a lot more questions now. So, hi, Min, thanks for the great talk. I'm curious, what is the cause of the sharp dip of adenosine right after VGLUT2 neuronal stimulation? A lot of adenosine A2A receptors in this. Okay, yeah, sorry. So that's the first one.
Yeah, I understand the question. Is you have a very sharp eye. Yes. So we didn't talk too much about this dip in the paper. So the short answer is this this dip is not adenosine, is adenosine independent, because if we use pharmacological method to block the A2A receptor, because the A2A receptor basically the adenosine sensor is a chimera A2A receptor. It can still respond to the A2A antagonist. If we give the A2A antagonist, basically we cannot measure any signal for using the probe. But this dip is still there after we apply the A2A or the sensor antagonist, which means this dip is adenosine independent. So we don't know exactly what caused this dip, but we have a theory. We think basically this is an intracellular sensor. The GIP is inside the cell. So when you have a protein inside the cell, you will respond to the pH change of the cell. So when you have a lot of spike, which is the case when we activate the glutamatergic neurons, so basically you will decrease the pH of the intracellular space. When you have a decrease of the pH, the decrease of the pH will dim the fluorescent protein. That's what we think is the cause of this dip.
That's really neat. So we have a couple more questions coming in. So
Sure.
One of them is a lot of adenosine A2A receptors are in the striatum. Have you looked at adenosine during the sleep cycles in the striatum, I think is the question.
Yes. We have actually measured adenosine level in many brain regions, including the striatum, the dorsal striatum, and also the ventral striatum, including the NAC. So basically, this kind of region, adenosine, I think they are high during wakefulness and REM sleep. I think the explanation is extracellular adenosine is tightly coupled with the neural activity in many cases. So in this kind of brain regions, basically the neural activity is relatively high during wakefulness and REM sleep. That's why we get a lot of adenosine during this period.
All right. So what is the next question is, what is the effect of adenosine in REM state?
We don't know. Yeah, exactly. We don't know. We have no idea because, as I said, there are a lot of ways to generate adenosine, but in the normal case, the physiological condition is tightly coupled with the neural activity. So during REM, the neural activity is very high. I think a lot of the time is even higher than wakefulness. That's why you have a lot of adenosine released by the cell. Yeah. This question is equivalent to what was the use of the high neural activity with REM sleep, right? We still don't have a clear idea. Yeah.
Oh, that's great.
That's a good question. Yeah.
So we have another question is, what is the interpretation for the differences from the chat and VGLUT2 neurons? Why do you think the cholinergic neurons have less effect?
So this is basically based on two experiments. One is the optogenetic activation experiment. When we use the 10 Hz, so basically a lot of, I think this is the maximal average firing rate of the cholinergic neurons during the physiological condition in the basal forebrain. It can cause very less adenosine release. But instead, if you use the average firing rate of the weak glu neurons to activate the glutamatergic neurons, you can cause a huge increase of the adenosine. So that means these two kind of cell and the physiological firing rate, the ability for these two cells to drive the adenosine release is very different. The cholinergic neuron has a very small contribution. The glutamatergic neuron has a larger contribution. On the other hand, when we lesion the weak glu neurons, we cause a significant decrease of the extracellular adenosine. This result really suggests that the glutamatergic neuron can play the major role in controlling the adenosine dynamics during the sleep-wake cycle. Yes. So based on these two results, I think we conclude that the glutamatergic neuron has a larger role than the cholinergic neurons.
Okay. I have two more questions. One of them is question about how adenosine pressure is released during sleep. So I think how does sleep basically lower your adenosine level?
That's a very good question. So basically, so in this paper, we mainly addressed how adenosine was generated. We don't use although during my talk, we use a lot of adenosine release, but we don't exactly know how, from my work, we don't exactly know how the adenosine was released by the cell. But according to a collaboration between my lab and the Lee's lab, so basically they studied the cellular mechanism of adenosine release. So you can find this paper in the archive. So they found the ENT transporter is very important for the generation of for the release of the adenosine. Basically, I think, or I speculate, my speculation is that, so a lot of the ATP was used inside the cell. So when you use this ATP inside the cell, you will build up adenosine inside the cell. And when the adenosine level is high intracellularly, so the ENT, basically this ENT can balance the concentration of the adenosine inside the cell or outside the cell. When the intracellular adenosine level is high, it transports everything and the adenosine towards outside the cell. So when the enzyme then can synthesize the ATP, use the intracellular adenosine. So this adenosine, so it creates a low adenosine level in intracellular. Then it will transfer the extracellular adenosine back to the intracellular space. Yes.
Okay, so I think we have two more. So what makes BF-SOM neurons unique to fire during sleep? What neurotransmitters are important for sleep regulation, somatostatin, GABA, or some other neuropeptides? That's a lot.
Currently, we don't exactly know. So for your specific question is what makes the SOM neuron firing selectively during sleep, some of the SOM neuron. So that's another thing why we want to do the single cell RNA-seq. We think in this, our preliminary evidence suggests that on the SOM neuron that they express the inhibitory muscarinic ACH receptor. So that makes it being inhibited during wakefulness, then during sleep, when the tone of the acetylcholine is lower, so the cell can fire. Yeah, that's our preliminary result. Right. And the second question, what neurotransmitter is important? Yeah. So that's kind of, I don't know. I think everything is important, yes.
All right, so the last question is, do you know if adenosine signaling regulate directly the glymphatic system? It is known that sleep promotes glymphatic, and I'm going to spell it out, G-L-Y-M-P-H.
Oh, I know, glymphatic.
Okay.
I don't know right now, yes. Yeah. So, because according to Dr. Nedgard, I mean, the glymphatic system is basically generated by the astrocyte, right? So I think astrocyte has been implicated in the literature. It has a close relation with the adenosine signaling. I think it could be possible that adenosine can have some role in regulating the glymphatic system.
Well, that is all of the questions. If you have any more questions, I'm sure that Minshu would be happy to answer them via email or a discussion. Again, thank you all so much for participating in the webinar, and we hope to see you in a week or so for our next webinar series. All right, so
okay, thank you.
Good night or good morning to all of those in the Pacific. All right, take care.